In this video, we'll talk about class 1 and class 2 MHC molecules. We'll look at their structure, their function, where they are expressed, and how they are presented on the cell surface. So welcome to Handwritten Biology, where biology makes sense when we draw it. So here is a lecture outline. But before that, don't forget to hit that subscribe button and tell your friends about us. First, we'll talk about what are major histocompatibility complexes. Then we'll talk about what are the structural difference between class 1 and class 2 MHC molecule. Then we will talk about the polymorphism in MHC gene expression and how that account for diversity. Lastly, we'll talk about the pathways and the cellular processes responsible for class 1 and class 2 MHC presentation on the cell surface. So stay tuned till the end. So major histocompatibility complexes are molecules which are essential for the immune system's ability to recognize foreign molecule or sometimes self molecule. So these are roughly the structure of the MHC molecule. Where can we find these molecules? We can find them on immune cells such as macrophages, dendritic cells, B cells, but we can also find them in any nuclear cells such as hepatocytes or epithelial cells. Big question is why major histocompatibility complexes are present on surface of the cell. What is their function? And what if they are not present on the cell? What functional ability of these cells would be compromised? So let us understand these in details. So class 1 MHC is present in any nucleated cell, whereas class 2 MHC is present in antigen presenting cells such as dendritic cell, B cell, and basically macrophages. Class 1 MHC has different chain organization. It has a alpha chain which is subsegregated into alpha 1, alpha 2 and alpha 3 subunits, and there is a beta 2 microglobulin which is produced by completely different portions. And this class 1 MHC displays endogenous antigen or sometimes viral peptide or tumor antigens. We'll discuss that in details. Class 2 MHC molecules displays exogenous antigen, maybe an antigen which is part of a bacteria's flagella or plasma membrane or even a bacterial component. So class 2 MHC molecules are having different structure. It has one alpha chain and one beta chain. Alpha chain has alpha and alpha 1 and beta alpha 2 subunits. Beta chain has beta 1 beta 2 subunits. So question is, how can class 1 and class 2 MHC displays antigen on the cell surface? What is the intricate nitty-gritty cellular details? So this is the structural difference of class 1 and class 2 MHC molecule. The difference lies in the peptide binding groove where the peptide is bound to the class 1 MHC molecule. So you can see the top view of the class 1 and class 2 MHC molecule, and you can see how class 1 MHC and class 2 MHC molecule can accommodate different size of the peptide. Now, class 1 MHC and class 2 MHC are produced by specific genetic clusters. So these are known as HLA clusters. There are MHC1 cluster, MHC2 cluster, and MHC3 clusters. Now, all of this is present in the human chromosome 6 P arm. The MHC gene is highly polymorphic. That means it has significant genetic diversity within a population. And this is an important thing to remember, that they are highly polymorphic and they exist in multiple allelic forms within a population. And these allelic variations result from genetic variations in the MHC gene. It turns out the variation is not everywhere. The variation is in specific amino acid positions, which are known as hotspots, where there is maximum variability. And this accounts for the diversity of the antigens that can be presented on the surface of the MHC. Imagine that one particular MHC is not variable, then it has limited potential to display wide variety of viral or bacterial peptide on its surface. That is not good for the immune system, right? It needs diversity, and that is achieved via this MHC gene polymorphism. Now let's talk about the MHC gene presentation. Let us quickly go through this process in brief and then we go into details. So first of all, MHC1 or MHC2, both are encoded by the genes. So they would be encoded from the MHC cluster. So MHC1 eventually moves to the endoplasmic reticulum, where the unfolded protein gets folded up with the help of certain chaperone proteins. Then beta 2 microglobulin associates with that. Eventually, using a transporter called TAP, all the endogenous peptides which are broken down using proteasome enters the endoplasmic reticulum. That eventually gets displayed on the surface of class 1 MHC. Now, using the cellular vesicle trafficking machinery, anterograde transport mediated presentation of class 1 MHC molecule happens. Now, in terms of class 2 MHC molecule, things are a bit different, because initially class 2 MHC molecule has basically no exogenous peptide in the endoplasmic reticulum. The exogenous peptide come and gets loaded onto the MHC molecule in the some sort of like endosome. So endosomes are formed when certain exogenous pathogens or exogenous proteins etc. get phagocytosed by the cell. They form early endosome and late endosome. This late endosome fuse with the vesicle containing class 2 MHC molecule, and then these exogenous peptides are loaded onto the top of the MHC and displayed on the cell surface. We can clearly see the difference between two. Now let us look at the nitty-gritty details and try to appreciate the process. So we have to understand endogenous antigens are presented by MHC1, exogenous antigens are presented by MHC2. Now again we are looking at MHC1 presentation. From the MHC cluster there would be proteins which would be translated onto the by the ribosomes present on the rough endoplasmic reticulum. They would enter the rough endoplasmic reticulum and would be folded with the help of chaperones like calnexin and ERP57. So ERP57 also helps in forming the disulfide bonds. Then beta 2 microglobulin eventually binds this complex, and this is a signal for these calnexin and ERP5 to dissociate. Next, another chaperone known as calreticulin binds to the class 1 MHC and form a complex with ERP57, tapacin and the TAP transporter. All of these together is known as the peptide loading complex, because it would load a endogenous peptide into the groove of the MHC. Now what happens is there are endogenous proteasomes which are known as immunoproteasomes. They break down endogenous peptides which get into the endoplasmic reticulum with the help of the TAP transporters. Now tapacin and all this complex help to load the MHC with an endogenous peptide. Then MHC moves from ER to Golgi and follows the anterograde pathway to be presented onto the cell surface. This is how MHC1 presentation happens. Now let's talk about MHC2 presentation. Things are different. Again, MHC2 cluster give rise to MHC. Things get translated and eventually the unfolded protein goes to the endoplasmic reticulum. There the folding happens, but at the same time we have to remember there is an invariant chain which is marked here in blue. This kind of blocks the peptide binding group. And even though there are endogenous proteins present in the endoplasmic reticulum, that is not loaded onto the top of class 2 MHC molecules. Now this is like kind of like a packaging material blocking a cup, let's say. So this is how it is nicely packaged. Eventually it moves from the ER to the Golgi, and MHC complex is routed through the Golgi to the overall anterograde pathway. Now invariant chain is degraded eventually when there is a change in the pH. Now meanwhile there are exogenous antigens which are endocytosed, and these endosome fuse with the vesicle containing class 2 MHC molecule. There are molecules like known as HLA-DM marked here in pink that help to remove these clip peptide and load that peptide binding groove of MHC2 with the exogenous antigen. So obviously now we can see the exogenous antigen displayed MHC2 molecule would be now presented on the surface of these cell, and this is happening only in a dendritic cell or macrophage or B cell, very selectively to the antigen presenting cell. Not everyone has this capability to present exogenous antigen. So I hope this was quick enough and understandable. Now we would quickly look at the differences in a tabular format. You can also take a screenshot. These are all the differences that we talked so far. Now, if you want notes that you can possibly download and practice MCQs, definitely follow this link which is provided in the description. And see you in next video. Don't forget to subscribe and tell your friends about our channel. See you in next video.